Hi there. Welcome to Hipolit's Biotech Breakdown. I'm Hipolit, and I'm the biotech expert at Dragon Gate Investment Partners. In today's video, I'll cover AlphaFold. So, what is AlphaFold? What's the protein folding problem? Why is protein folding so important? Did AlphaFold really solve the protein folding problem? In this video, I'll answer all these questions and provide an overview of the AlphaFold software. But first, it's important to have an understanding of proteins. Not that kind of protein, but the proteins that underpin the biological processes found in the body. Nearly every function that our body performs relies on proteins and how they move and change. Proteins are essentially chains of amino acids that, when straightened, resemble a string of beads. Even though there are hundreds of amino acids found in nature, only about 20 are needed to make all the proteins found in our bodies. The unique characteristics of proteins are determined by the amino acids they contain, how these amino acids are arranged in a chain, and the further complex interactions that this chain makes with itself and its environment. Proteins have a unique shape and three-dimensional structure that allows them to perform a precise function. By understanding this structure, scientists are able to work on new medicines to tackle disease and even potentially unlock some of the mysteries of life. You may be wondering, why is protein folding so important? Many people are suffering from diseases either tied to or caused by protein misfolding. These diseases include Alzheimer's, cancer, prion disease, and type 2 diabetes, just to name a few. By understanding a protein's structure, scientists will be better equipped to understand and tackle disease. For decades, scientists have been working to find a reliable method to determine a protein's three-dimensional structure just from its sequence of amino acids. This challenge is known as the protein folding problem. Figuring out the exact structure of a protein remains an expensive and time-consuming process, meaning we only know the exact structure of a small fraction of the proteins known to science. Computational methods to determine the exact 3D structure of a protein from its sequence of amino acids have proceeded along two complementary paths. These paths focus on either physical interaction or evolutionary history. Physical interaction programs heavily integrate our understanding of the molecular driving forces of proteins into either kinetic or thermodynamic simulations of protein structure or statistical approximations. In recent years, the evolutionary approach has provided an alternative in which the constraints on protein structure are derived from the bioinformatics analysis of the evolutionary history of the protein, similarity to solved structures, and pairwise evolutionary correlations. Despite advances, physical and evolutionary history-based approaches have yielded predictions that are far short of experimental accuracy. This is in the majority of cases in which a close homologue, or common ancestor, has not been solved for through experiments. This has limited their utility in many biological applications. This is where AlphaFold comes in. AlphaFold is an AI program created by DeepMind that can accurately predict and model the three-dimensional structure of proteins just from a sequence of amino acids. Interestingly enough, DeepMind is the same company behind AlphaGo, which in 2016 became the first computer program to defeat a professional human Go player. In 2016, DeepMind also started the AlphaFold project as a way to combat the protein folding problem. AlphaFold is taught by learning from the sequences and structures of around 100,000 known proteins. The latest version, also known as AlphaFold 2, can predict the shape of the protein at scale within minutes down to atomic accuracy. For protein folding, this is huge. AlphaFold greatly improves the accuracy of structure prediction by incorporating its novel neural network architecture and training procedures based on the evolutionary, physical, and geometric constraints of protein structure. You may be wondering, how does AlphaFold work? AlphaFold could be thought of as three essential parts: pre-processing, evoformer, and the structure module. In pre-processing, the inputted amino acid sequence is used to pull information from external databases. This information is then mapped onto pair representations and a multiple sequence alignment, or MSA for short. This MSA is thought to be evolutionarily related to the target. In terms of proteins, MSAs are generally the alignment of 3 or more protein sequences of similar length. From this output, similarity can be inferred, and the evolutionary relationships between protein sequences can be studied. Pair representations are tools used to identify regions of similarity that may indicate functional, structural, or evolutionary relationships between two biological sequences. In the next step, evoformer takes and repeatedly updates the MSA and pair representations until there is enough information about the relationship between amino acids. Finally, the structure module then takes the information gathered from evoformer, transforms it, and maps it in the three-dimensional space. This structure is then run through a relaxation phase to fix any violations, and the end user is left with a ribbon diagram of the protein along with 2 confidence metrics. The 2 confidence metrics include predicted LDDT and predicted align error, or PAE for short. Predicted LDDT measures the percent of correctly predicted interatomic distances between amino acids. In this metric, a higher score indicates a higher confidence in the local structure. PAE is the expected position error of amino acid X if predicted, and the true structures aligned on amino acid Y. In this metric, a lower number indicates a higher confidence in the protein's domain. You may be wondering, so where can I access AlphaFold? Well, DeepMind has made AlphaFold available in 3 places: the source code, the collab notebook, and the protein structure database. The source code is the most powerful tool of the 3, as it offers the most precision and customization. One important thing to note about the source code is your computer needs to be able to handle the 2.2 terabyte file. The collab notebook is a useful tool for running individual sequences and should offer accuracy close to the source code. The collab notebook does not require any coding experience or an installation on your computer, as it's cloud-based. It's important to note that this is not recommended for large prediction jobs and may be unreliable for long sequences. Also, be prepared for long wait times, especially when the program is searching large genetics databases. AlphaFold's protein structure database is offered in partnership with EMBL's European Bioinformatics Institute. In its initial release, the protein structure database covers all 20,000 proteins in the human phenotype, as well as the proteomes of several other biologically significant organisms, from E. coli to yeast and the fruit fly to the mouse. While AlphaFold has shown promise, it's important to be aware of its limitations. Interactions between partner proteins and multimers are not currently in its database. Also, the AI program does not currently predict other important aspects of protein structure, such as metal ions, cofactors, and other ligands. It is also important to note that proteins are dynamic systems that change their shape depending on their environment or state within a functional cycle. While AlphaFold has not completely solved the protein folding problem, it has offered a practical solution and made a huge leap within the field. With this leap, there's a huge amount of excitement around AlphaFold as it opens the door to tackle broader questions and offers a solid base to build off of and experiment with. Thank you for tuning in to Hipolit's Biotech Breakdown, and for more information on this topic, please check out my PowerPoint in the description.